Okay, so welcome to the 13th meme seminar. Today we have Ülkü Uzun, I think. She is originally from Turkey, and she did her undergrad in molecular biology and genetics at the Middle East Technical University in Ankara. And then after that, she did meme, and then after meme, she did her PhD in the Department of Biochemistry at Oxford, and she just graduated. And so she's currently looking for a postdoc. And she will be talking to us today about some of her PhD work. So take it away.
Should I share my screen? Yes. So yeah, hello everyone. I am Ülkü Uzun, and I was from the 5th meme cohort. And very recently, I finished my PhD in Jane Mellor's lab at the University of Oxford. And I worked on the regulation of RNA polymerase II transcription by Spt4 in budding yeast. So I prepared not to just directly start with my project. I prepared this slide, although Charles a little bit talked about it. So I'm from Turkey, and I did my undergrad studies in Ankara at Middle East Technical University. And I was always interested in regulation of transcription and translation, and I did a couple of projects related to these topics. And then I did my meme, and I've been at 4 different universities in my 4 different semesters. I started in Uppsala, then I moved to Montpellier. I did my master thesis at LMU, and then I finished off in Boston. And when I was doing the meme, I always wanted to take more molecular approaches, and I focused on some genes and proteins regulating prezygotic and postzygotic speciation in different organisms. I also worked on ribosome evolution and how it affects cell growth development, how it limits cell growth in bacteria. And finally, I did my PhD at the Department of Biochemistry in Oxford, and I worked on transcription regulation. So today I will mainly talk about my PhD projects. I will first tell you a little bit about transcription and how it is challenging for transcription to go through chromatin. And then I will talk about transcription elongation factor Spt4, what we know about that, and why I chose this protein to focus on. And then I will introduce methods and results as we go through the presentation. And then I will summarize and tell you a little bit about open questions in the field. So in eukaryotic cells, RNA polymerase enzymes transcribes RNAs from DNA template. And there are a couple of RNA polymerases, but the main focus is usually around RNA polymerase II, because it's the main transcribing for messenger RNAs, which are then translated into proteins in cytoplasm. And here I wanted to tell you a little bit about different stages of transcription mediated by RNA polymerase II. So if you imagine that a gene transcription start site is this point, I hope you see my pointer, and the end of gene is here, there are different sets of factors. They are either interacting with DNA elements or they're interacting with polymerase II, and they regulate different stages. They recruit RNA polymerase II to the initiation site, then they help Pol2 elongate through transcriptional units along the gene bodies, and then they regulate termination, meaning that the mRNA, native mRNA is released and Pol2 is falling off the DNA template. And what we know is initiation of transcription and the factors helping Pol2 transcription, more or less well known at the initiation stage, but we do not know a lot about elongation and termination stages. And in my lab, in James' lab, we were focusing on elongation phase. And here, for simplicity, I just showed DNA as this one straight line here. But in this, it's not real, of course, like this is a big lie, because in real life, in eukaryotic cells, DNA is not found in a straight linear form, but it is found in a compact form, which is known as chromatin. And chromatin is formed by nucleosomes, which are building blocks of chromatin structure. And this formed of 148 base pair DNA wrapped around 8 histone proteins. And the interaction between histone proteins and DNA limits the movement of processes on DNA template. So when any enzyme needs to reach to DNA, nucleosomal DNA, this formation kind of sets physical barriers. And I would like you to remember this 140 base pair because it will later be important for my results section. And then if we go back to my initial scheme, if I put this nucleosomes, it would be more realistic to show in that way that while Pol2 is transcribing, it actually interacts with some nucleosomes. And actually there are, in last couple of years, there have been amazing studies, structural studies. And one of the models I put here to be more realistic. So on the right, this gray big globular protein is RNA polymerase II. And as you can see, during transcription, it tries to go through nucleosomal DNA. So we know that there are already different sets of protein groups regulating chromatin. These can be ATP-dependent chromatin remodeling complexes, which are actively, by spending energy, which are actively evicting or sliding or exchanging nucleosomes. Or there can be histone-modifying enzymes, which are adding like phosphorylations or acetylation marks, that kind of stuff. And in the end, it can affect the DNA, nucleosomal DNA and protein interaction. And there are also histone chaperones, which are binding to free histone pools in the cell. So they regulate nucleosome assembly or disassembly during like cell regulation in different pathways. So we know a little bit about transcription. We know a little bit about chromatin. What's not known is how transcription, like during transcription, chromatin template is regulated. I say like we don't know, we started to investigate that in the last 5 years. It's been really a hot topic. And besides proteins that I listed here, like protein groups, as I told you before, there are also transcription elongation factors, which are interacting with Pol2. And we know that some of these factors might be important for helping Pol2 to move through chromatin template. And there are many, many proteins. And amongst them, I chose Spt4. This is just one of the proteins. And now I want to explain a little bit what we know about this protein and why I chose this protein. Spt4 is a transcription elongation factor, which means that it engages with Pol2 during transcription. This Spt4 has been discovered almost 40 years ago. And while it was discovered, other Spt proteins are discovered as well. One of them is Spt5. It's the binding partner of Spt4. And here I put a structural studies just came out 2-3 years ago. This is yeast studies. What we know is Spt4 and Spt5 together sits just in between RNA polymerase II and upcoming nucleosomal barrier. So physically, the position of Spt4 is very convenient to regulate Pol2 movement during transcription. And this complex, the Spt4, Spt5 complex, also known as the SAF complex in mammalian cells, and it is very famous. Like, go and open a publication. If they are talking about polymerase II elongation, they definitely mention Spt4/5 or the SAF complex, and they say that it's important for transcription, but we still do not know what does this protein do or the complex. One of the very outstanding feature of this protein is that it's one of the most highly conserved transcription elongation factor, and it has homologs in archaea and eukaryotes, human, Drosophila, mice, plant. So it means that like organisms with nucleosomes have Spt4. One of the other like knowledge that we know about this protein, the mutants of Spt4 cells share some similar phenotypes with histone chaperone mutants. And finally, we know some in vitro studies, biochemical studies that. So, and these studies are very recent. What they do is they purify polymerase II, they put DNA and nucleosomes together, and they check how Pol2 can transcribe over nucleosomal DNA. Without Spt4 and Spt5, Pol2 is having hard time to transcribe through this template, and it stalls at certain points. But when they add purified Spt4 and Spt5, Pol2 stalling is reduced very significantly, and it already shows that the function of this complex Spt4/5 might be important for transcription over chromatin template. So altogether, genetic interactions, structural studies, in vitro studies points to a role for Spt4 and 5 into transcription in chromatin template. However, there is no in vivo evidence, so we do not know what's really going on in cell. And usually, many studies focus Spt4 and 5 together as a complex, and like we are far from understanding the individual contribution of these proteins into transcription. So during my PhD project, what I wanted to answer is to define exact contribution of Spt4 in transcription, and I used budding yeast as model organism. I did several high-throughput sequencing techniques I performed, and I'm gonna go through them one by one while I'm introducing my results. But what I in general investigated is that if Spt4 impacts nucleosome positions, where does Spt4 localize across the genome over the gene bodies, and how does Spt4 impact transcription, and how does it impact RNA polymerase II distribution? So first, given that Spt4 seems to be very important for nucleosome interactions during transcription, I wanted to check if nucleosome positions overall the genome was changed upon Spt4 knockout. To answer this question, I used MNA sequencing. In this technique, we use this enzyme, special enzyme MNase, and MNase digests internucleosomal DNA. So upon MNase treatment, I get fragmented DNA from nucleosome protected regions. And when I isolate this DNA, and when I sequence and align to yeast genome, I infer the position of nucleosomes from protected DNA. So for example, this is gene 1, and this is transcription start site, and this peaks defines the position of nucleosomes. And from the transcription start site, we number, for convenience, we number the nucleosomes +1, 2, 3, and so on. And on the other side, the nucleosome is called -1. So as a result of DNA sequencing, we get information from many genes, and in yeast there are several thousand genes. We do this for all the genes, so we do genome-wide sequencing. And then when I computationally align all the genes from transcription start site, and when I take the average of the signal coming from the nucleotide positions, I get average genome profiles, which we call metagene profiles, and I'm going to be showing you several of them today. So I wanted to explain this. So this is the overall pattern of the position of nucleosomes coming from several thousand genes. So yeah, what did I do is I applied this experiment to wild type and to Spt4 knockout cells. And when we look this average gene profiles, what we see in black is that the position of nucleosomes in normal cells, and in blue is position of nucleosome in Spt4 knockout cells. So as you can see, there's a shift towards the 3' end, towards the end of the genes. And when we do more detailed analysis, what we see that the position of +1 nucleosome was actually not altered in between two conditions. And when I say position of +1 nucleosome, I am talking about the center point of +1 nucleosome relative to transcription start site, the distance. But actually what we have seen is the distance, the position of +2 and downstream nucleosomes were altered. So in the end, this experiment showed me that without a change in the position of +1 nucleosome, the spacing between nucleosomes were increased in Spt4 knockout cells. And like the analysis also showed that this is significant for +1 and +2 nucleosome, +2, +3, it's still significant for downstream nucleosomes. But because of technical reasons, I mean, normally with this technique, the downstream nucleosomes, we do not get very perfect phasing. So it's usually more convenient to talk about first 4 or 5 nucleosome positions. So what we know as a result of this experiment is that Spt4 is required for normal nucleosome positioning. And knowing that +1 nucleosome is not very much affected already suggests that maybe the very beginning of transcription is not much affected, but the downstream processes changed in Spt4 knockout cells. And next, I wanted to understand the localization of Spt4 over the gene bodies. And for this, I need to explain you a different technique first, and I'm going to explain what technique I used to answer my question. So there is a technique called native elongating transcript sequencing, and this technique is used to map exact position of polymerase II. So during transcription, RNA polymerase II, DNA, and native RNA are really strongly attached together. What we have is, we have flag-tagged RNA polymerase II, and we immunoprecipitate it. And when we immunoprecipitate Pol2, we also immunoprecipitate native RNA. And then what we do is we digest protein, we digest DNA, and then we isolate RNA attached to Pol2. And then we do 3' end sequencing. This is very important because when we do sequencing from only one side, we can understand the very last nucleotide added by Pol2, and that would give me the position of nucleosome at the time of harvest. So if I know the last nucleotide added, I can understand if my Pol2 is at the beginning of gene, at the middle, or in the end of the gene. So this technique, NetSeq, is used for mapping position of Pol2. And in our lab, a couple of years ago, another technique has been developed, very similar to NetSeq. But in this technique, instead of polymerase II, we flag tag elongation factor, and again, we immunoprecipitate. So if the factor is associated with Pol2 at the given point, then we get to have a signal from this transcription elongation complex, so that we can understand the elongation factor was engaged with Pol2 around that point. So I did map the position of Pol2, normal Pol2, and then I mapped the position of Spt4 and also Spt5 because they're in a complex and I expect them to move together. And on the right, I wanted to give you some examples, and I also wanted to show you that I did these experiments in duplicates and they are quite consistent. And what we see is wherever we see Pol2 signal, we usually see Spt4 and Spt5 signal as well. On the left, when we look at the genome-wide genome averaging plots, what we see is in pink and in blue, the pattern of Spt4 and Spt5 is very similar to Pol2 in black. And with this experiment, we understand that Spt4 and Spt5 joins transcription very early. They seem to travel with Pol2 because we get signal throughout the gene body. But at the end of the transcription site, which is polyadenylation site, we do not get very much signal from this factor, so probably they fall off Pol2 around this point. So with this, I understand that yes, most of transcription elongation phase, Spt4 and Spt5 is engaged with Pol2. But I also wanted to understand if the association of Pol2 was constant throughout transcriptional unit, or at some regions, the Pol2 or Spt4 interaction with Pol2 was more engaged or increased or depleted. To understand that, what I did is I divided Spt4 engaged Pol2 signal by total Pol2 signal to see where Spt4 was enriched or depleted. And very interestingly, when I did that, I see these faces. And these faces looked very much like, this is surprising, because if these factors are always with Pol2, I expect to see a straight line. But I started to see these faces, which looked very much like nucleosome faces. And to see that if that was really related to, associated with nucleosome positions, instead of aligning the signals to transcription start site, I aligned them to center of +1 nucleosome to get more smooth, to see association better. And then what we see is, by the way, now it's the time that to remind you that the nucleosomal DNA was 147 nucleotide. So I highlighted something close to that number, 140 nucleotide here. So the first 70 nucleotide is the first half of the nucleosomal DNA. The dashed line is center of nucleosome, and the second half is the second half of nucleosome. What we see is, very interestingly, the association between Spt4 and polymerase II was depleted in the first half of nucleosome, but it was stronger in the second half of nucleosomal DNA. And this was also true for Spt5, as we expect from a complex. So with this experiment, what we've seen is Spt4 seemed to oscillate on and off Pol2, and this is based on nucleosome positions. And this might, like the position and association of elongation factors with Pol2, might also suggest that these factors are maybe doing something related to transcription through nucleosomal DNA. And very finally, of course, I want to understand how Spt4 impacts transcription. And to answer this question, we map the position of polymerase II, which reflects transcription, by using the technique I explained a couple of slides before. And to answer this question, again, we used wild type and Spt4 knockout cells. So again, I wanted to show you some single gene examples. On individual genes, what we see is the signal, Pol2 signal coming from Spt4 knockout cells were increased at the beginning of genes compared to wild type cells at single gene examples. And across the genome, we also see the same pattern. We get high signal at the beginning of the genes, which means that like there is more Pol2 signal coming from around this region. And of course, we want to understand why there's more Pol2, because that will answer our question about what does Spt4 do. And to answer this question, we collaborated with mathematicians. I will not go into details of the model. I will just give two big predictions of the model. So one of the predictions was that if there is higher initiation in Spt4 knockout cells, we would get more signal from the beginning of the genes, which is also very intuitive. To independently test this prediction, what I did is I looked at this protein called SUA7, which is a transcription initiation factor. SUA7 normally goes to promoter side, like beginning of genes, even before Pol2 arrives there. And it is a subunit of pre-initiation complex. And when I assess the level and the position of this protein, I see that between wild type and Spt4 knockout conditions, there is not much difference. So my experimental data does not support any change at initiation stage of transcription in Spt4 knockout cells. And the second prediction was that if there's some problem with elongation, especially at the beginning of the genes, if there's slow elongation, Pol2 would accumulate there, and that would create high Pol2 signal. And of course, we already know that nucleosomes are physically barriers to transcription. I wanted to check if the high Pol2 signal was correlated with nucleosome positions. And as I did before, I aligned my net-seq signal to nucleosome positions. And very interestingly, what we have seen that the high signal of Pol2 was actually within the first half of +2 nucleosome. We saw some mild effect around other nucleosomes as well, but it was very significantly high here. And of course, when I see that, it's very, like, to me, it's a striking result. But at the end of the day, what I do is I delete Spt4 completely out of the cells. And I do not know if this effect I'm seeing is just directly because of Spt4. To understand that, I also wanted to deplete Spt4 protein in the cell. And I used the method called Ankara. With this method, we have special cell construct. And with the addition of rapamycin, we can drag protein out of nucleus to cytosol. So when there is no rapamycin, we get this Spt4 signal, which is GFP tag in the nucleus. And upon rapamycin addition, in 2 hours, we start to see these hollow regions within cells, which we understand that Spt4 is depleted from nucleus, and it's in the cytoplasm. I also quantified this signal by using chip qPCR, but this is more convincing and nicer to see, so I wanted to show this. So I know that from my quantification, 85% of Spt4 was depleted from the cells. And what did I do is I did map the position of Pol2 again upon Spt4 depletion, which is in orange here. And very interestingly and convincingly, what we've seen is the change in the distribution of Pol2 was again around +2 nucleosome compared to navy control cells here. So with this in two different backgrounds, we show that in the absence of Spt4, RNA polymerase II tends to accumulate in the upstream phase of +2 nucleosome. So in general, what I showed today is that Spt4 is important for nucleosome positioning. The interaction of Spt4 with polymerase II changes based on the position of nucleosomes. And in the absence of Spt4, we get to see accumulation of Pol2 within the upstream phase of +2 nucleosome. So altogether, our model is that in wild-type conditions, Spt4 somehow regulates movement of Pol2 through the +2 nucleosomal barrier. And in the absence of Spt4, this mechanism is compromised and Pol2 starts accumulating around this region. So one of the very exciting things is that these in vivo findings are in line with structural, genetic, and in vitro studies. But on top of that, it shows that this might not be true for all cases, and Spt4 function might be more crucial. It seems to be more crucial around +2 nucleosome, so in early elongation phases rather than within gene body. So there might be other proteins regulating transcription through +3, 4, 5 other nucleosomes, but Spt4 seems to be crucial for Pol2 transcription over nucleosomal, +2 nucleosome barrier especially. So one of the open questions is, which is, it will be really interesting to see in the future, if Spt4 regulates Pol2 movement by interacting directly with histones. For some proteins, we know that this is the case, or it might be interacting with nucleosomal DNA. There are some models, in vitro models, showing that Spt4 or Spt5, if they interact with nucleosomal DNA, they might just keep it away from nucleosome so that Pol2 can pass more easily. So that might be the case. There might be other factors still Spt4 interacts with, and it fine-tunes the movement of Pol2. The other thing is, I told you from the beginning that Spt4 is very conserved protein. And of course, it will be very interesting to see if this is conserved in humans, because Spt4 and Spt5 might be interacting with other transcription factors like MYC, which is involved in cancer and so on. So it would help us understand mechanisms better. And also, one of the things I didn't mention at the very beginning, Spt4 is an important and famous factor in human cells because it's involved in a process which is called promoter proximal pose, which is in mammalian cells, Pol2 poses at the beginning of genes, which does not exist in yeast. But it would be really interesting to see if this function of Spt4, the function of Spt4 modulating Pol2 movement around nucleosome barriers, if it is related to promoter proximal pose. And of course, today I only focused on one protein, but this field is just kind of, two fields are not new, but people started bringing these two fields together just recently. And of course, the big question is what we really want to understand, that how to achieve efficient transcription over the nucleosomal DNA. So with that, I should really thank my supervisor, Jane Meller. Harry Fischel was a postdoc in our lab when I started. He showed me how to do NetSeq, TEFSeq, and analysis of that. Tom Brown and Andrew Angel. They did mathematical modeling. In the research, it's already on bioRxiv. We submitted, but you can always, I'm happy to have your questions now. But if you have more comments and questions for later, feel free to contact me. I'm always more than happy to talk about my project. And yes, that's the end. So I can see a hand, but should I do something or is it charged? Am I done?
That was super cool. You can take the questions. I think Azia has her hand up.
Yeah, hi, Ülkü. It was super nice presentation. So I don't know if I missed it, but I was just curious. In one of your slides, you showed this GFP expression of your protein, and I was wondering what is the connection between rapamycin and the protein, and how it's helping for the visualization of your GFP. Just like a mechanistical question, but I was just really interested.
Oh, it is just, so the thing is, it is Ankara way. It's a different system to deplete proteins. So this is a yeast protein depletion mechanism. Plant protein depletion mechanism. So my Spt4 protein has a special tag.
Yeah.
It also has ESP, just to monitor localization of my protein.
Sorry, yeah. Can you repeat the last sentence?
So my Spt4 protein has a special tag. And then it also has GFP tag. So this special tag, which is called FRB on my Spt4, in my yeast cell construct, it interacts with some other protein. When I add rapamycin, they find the ternary complex and they shuffle, they export Spt4 to cytoplasm. Actually, I have ribosomal protein because ribosomal proteins are always shuffled from nucleus to cytoplasm. So when I add rapamycin, because of this special construct, when I add rapamycin, my ribosomal protein and Spt4 binds together. So rapamycin glues them together, and then I can drag my protein out of the nucleus. And I also have, as I said, GFP tag on Spt4, so that I can monitor where I get the signal from, nucleus or cytoplasm. I don't know if this was the answer of your question. If it was clear.
So can you use that method to export any protein from the nucleus?
So the thing is, yeah, protein should be nucleus, like nuclear protein. I think some people tried the other way around as well, cytosolic protein to nucleus. But yeah, it can be applied to, this is I think this is a 10 years old technique now, and they have tried with so many proteins, and it efficiently works with many, many proteins. There are some proteins, like I tried with, for example, some important kinase proteins in cells, which are really essential genes. It doesn't work well with them. I don't know why. Cell has some mechanisms to protect themselves, I guess. But many proteins you can deplete.
How does it figure out which protein to attach to again?
No, I have already construct. So I already have special cells, which one subunit of a ribosome is tagged with a special tag. And then my target protein is tagged with another special tag. And when I add rapamycin, they come together. They also have some deletion in cells, like this, because this is usually this method works in plant cells, and they just integrated it into yeast cells.
So you order those tagged.
So you order the background strain, which comes with already system in it, and you only RPV tag your target protein to be depleted, and once you add rapamycin, it depletes your protein out of the plant.
Like the tag is like a protein molecular interaction.
It's, I think it is 12 amino acid long tag.
Okay, cool.
Yeah, and for some proteins, it might be more efficient. Like some people only does it for 30 minutes, 1 hour. In my case, I just saw the depletion upon 2 hours. So it needs some optimizations. But it's good to see because it's conditional, right? I grow my cells and I add and I see the immediate effects, what happens to cell. And it's also good for using some essential genes, which we did for Spt5. Spt5 is essential, but this is already published before I did my project, so it wasn't new.
How do you, can you explain the NET-seq again? I understood everything until the end.
So yeah, this is a, so let's see. So Pol2 has an active cleft, and it's like C-shaped protein. It has DNA in it, and even I have the structures, right? I can show. So it has this active site, and it has DNA in it, RNA in it when it is transcribing. And this is really hidden, just it's really strong structure. And I do not do any fixing or cross-linking, any chemical extra thing. I have this one of the subunit of polymerase II flag tag, and I do immunoprecipitation. And when I do immunoprecipitation, I pull down DNA associated within Pol2, I pull down RNA, other proteins as well. And then what I do is I do protein digestion, I do DNA digestion, but I keep RNA. So I isolate all the RNA. And then what I do is I add some adapter to the 3' end of the RNA, and then I do sequencing. This is from whole cell. And when I do the sequencing, I know the very last, like I know where is the end of
You know the position of the polymerase, but what is that? How does that tell you where the nucleosomes are?
Oh, it doesn't tell where the nucleosomes are. This is just the Pol2 position. Nucleosomes is different. So for nucleosomes, we use MNA-seq with enzyme. I digest all the DNA, and I have protected DNA showing me the position of. So yeah, there are a couple of high-throughput sequencing techniques, and they have, like, one of them is DNA, one of them is RNA.
And yeah, then what was the third one? The TEC-seq? Or
TEC-seq, it is transcription elongation factor associated net-seq. So if my factor is on Pol2, when I immunoprecipitate my factor, I do exactly the same for Pol2, but this time I pull down from elongation factor. So if there is no elongation factor, I do not get any signal around that position.
Got it. So then the difference between the two would just be whenever the polymerase has the elongation factor attached.
Exactly, and these are single nucleotide resolution. So I don't know how familiar you are with these techniques, but usually when they do these techniques, the precision is only 100 nucleotide. Like, it's like big chunks of DNA or RNA. They don't know where exactly proteins, but these are single nucleotide resolutions. So this is the highest definition you can ever get, that kind of stuff. And this is a pretty new technique. And it helps me to see this. Otherwise, you wouldn't see these phases. You would see just, yeah, protein is there when Pol2 is there. What I see is the association between my protein and Pol2 changes based on very specific locations.
Hey, Ülkü. Do you hear me well? Can you hear me? Yeah. Yes. How are you? Thanks for the presentation. It was super nice. I have one question, which is a bit different. So I know that histone modification marks are associated with silencing or expression of genes, but I'm not sure how actually molecularly that works and like if what proteins, what transcription proteins are associated with this. So I was just curious if you know about this.
Yeah, that's amazing question, and I am curious about that too. But the thing is, we do not like in terms of Pol2 passing these nucleosomal barriers. We do not know how histone modifications impact Pol2 movement. This is, as I said, like this is just something new. People started looking at it, and we do not know if there's phosphorylation on, or if it is silenced, or if there's more phosphorylation, if there's less, some of the modifications. We do not know how it impacts the movement of Pol2. In general, like in very broad sense, we know that some of the elongation factors are helping recruitment of some histone modifying enzymes. Some of them do not like them. They just like, because while transcription is happening, the nucleosomal DNA is unwrapped, and they, after Pol2 passes, it's rewrapped. And some of the modifications probably changes. So they have to first remove some modifications and put them back. That kind of regulations. But this is, again, it requires very much, a very high resolution. And there is no big study around this. We know that some histone modifications are increased during when transcription activation increased, but we do not know how it affects movement of Pol2. It might be just recruiting other factors as well. So the straight answer of question, I don't know.
Yeah, because it's really amazing, like how, like, depending on where the mark, in which amino acid the mark is, it can change.
It can change, and some modifications does not even affect this compaction.
Exactly. Yeah.
But yeah, it's I don't want to say, but it's a bit of a mess because there are so many data, but studies are not coming together. And also, just one of the things that I always want to tell when I introduce my results and other genomic high-throughput sequencing, we work with population of cells. So whatever I show you is that coming from 2 million cells. And I do not know exactly in which cell, like overall, it's convenient for me to say that where my Pol2 accumulates is the same cell where nucleosome is shifted a bit, but maybe they are different cells. Like, of course, overall response seems like that, but we do not know. So this is also like in each cases, in each round of transcription, modification changes, position changes. So I think there will be more studies in coming years. Like, not I think, I know there are a lot of studies.
So do you think like single cell studies would provide different or might provide different results?
Yeah, definitely. And I am very super interested. Like, for example, single molecule studies would tell me if DNA is interacting with my protein while Pol2 is moving, for example. I could see that one-to-one. And I know there are some groups working on that. But adding nucleosomes to single nucleotide studies are also another challenge. So they can do it on normal template. But it's, I think, a bit more challenging to add a couple of more complicated constructs, but they are doing it. They're coming to that point.
Cool, thanks. Very nice.
I see, but should I can see some hands, but I don't know how to unmute. Yes.
Can I ask? I'm curious if, well, you're showing the results on general for like genome-wide for all of the genes using this transcription start site, right? But have you looked at the difference between different genes? They may be developmental genes or some other genes will have different type of regulations. So the signal will be different based on the genes. If that was observed or not.
Yeah, that's an amazing question, and I think it's very fair to think that there might be some subset of genes having different reactions. So in yeast, we can not really talk about so much developmental genes because it's unicellular organisms, but of course there are different genes related to metabolism, like DNA damage and other sorts of pathways. But for these factors, when I wanted to find some gene subsets behaving differently, I just see that they all behave the same. Like they all accumulate POL2 in the absence of Spt4. And the amount of Spt4, because I show you the position of Spt4, it is also in line with the amount of total POL2 in the cells. So what I show you in yeast cells at least seems like it's happening for all transcription events. So this should be the general mechanism, but we couldn't find any specific types of genes behaving differently. I tend to see increased transcription in stress genes in the absence of Spt4, but this is also kind of expected because you remove one of the elongation factor and cells are in trouble, so they want to fight. But it doesn't change the overall phenotype that I see.
All right. Yeah, maybe also the signal somehow averages because of the population level cells.
Yeah, exactly. So I showed you today average signal, but when I do my analysis, and also it's also on the manuscript if you were interested, I also show it for across the genome with heat maps for single genes, and the response looks similar for several parameters. So it's conserved across the genome.
Okay, cool, thanks.
I think Azia had another question. It's in the chat. I can read it. Is Spt4 has any role in POL2 elongation speed? There are studies showing if you slow down POL2, we have longer lifespan.
Yeah, that's an amazing question. So Spt4 does have effect on the overall movement rate of POL2 because it poses more, but it doesn't have any effect on the addition of new nucleotide rate. So we did also, this was one of the parameters in our mathematical modeling. So Spt4 does not impact the addition of new nucleotides to RNA, but overall, the speed of, because it poses more, the overall speed of POL2 is slowed down. What was the second part of question? She says it increases overall lifespan.
It was more like a comment. So there, I don't know if you know, there is a lab, RNAPII lab in Cologne, and Partridge Lab. So they're very famous, this lifespan extension, labs, like they're doing, aging studies. And one of their papers was about that if you slow down the POL2 activity, like the speed of POL2, both, I think, in C. elegans, also in Drosophila, they saw that it actually causes lifespan extension. I think now they're like trying to find, like, I'm not sure, I didn't read it through, but there are still like some debates about the mechanism behind it. So I was just wondering if somehow the Spt4 has anything to do with the speed, and maybe that's one of the targets in the end.
No, but actually it's amazing because I mean, I didn't check it in terms of aging, but I know that the doubling time of the cells are increased. So they are slower. I don't know how it would translate to aging, of course. This is something that I haven't looked at. And yeah, speed of POL2 is also, I mean, we have some models, but we didn't do the technique to directly measure the speed of POL2.
Yeah.
There are some techniques to do that, and it would be interesting to see this.
Maybe, I don't know, checking the growth of the yeast, or I don't know, something like that.
No, I think there are some aging studies even in yeast. They just passage and they check some
exactly
stimulates and stuff. It would be really, I never thought about this aspect. But it would be
just an additional idea.
No, it's amazing. Like, it's also like, you know, when I was doing my project, I was like so much into transcription, but there's, it's related to everything, and it's amazing, very exciting. Yeah, thank you. No, I can have a look at this, definitely.
Good.
Okay, I have a basic question. How do the nucleosomes know where to go?
Amazing question. These are just like, you know, these studies are very recent. And I just love talking about them because they didn't know until recently. But now what they know is, so there's replication, right? And during replication, DNA, they have to like replicate DNA, and they have to remove all these nucleosomes, and they have to put them back. So there are some anchoring proteins. They go to, there are specific DNA sequencing. Anchoring proteins first go there and like marks, this will be my point where I start putting my nucleosomes. And then the next remodeler complex goes and add 1 + 1 nucleosome. And after +1 nucleosome, another complex goes and starts like, because DNA sequences has also some bias. Some of them are AT rich, some of them are like, they have specific sequencing for other factors to go and bind. And the position of first nucleosome is decided by different remodeler complex, and the spacing between nucleosomes are decided by another complex. So they first add one anchor protein, and taking it as a reference, they just align other proteins, other nucleosomes around them. Yeah, and that's what we know so far. But yeah, there are a lot of protein. And when I showed you that the position of nucleosomes are changed in Spt4 knockout cells, it's just by 4 nucleotides. And if I didn't know anything about it, I would be like, so what? 4 nucleotides, like it might not be that important. But indeed, even when you delete some of the chromatin remodelers, sometimes you do not see an effect because these processes are very tightly regulated. They are very redundant, so robust. So having even a 4 nucleotide difference is a big issue for the cell. And also highly transcribed genes has more changes compared to lowly transcribed genes. So it's different. I don't know, I told you more than your question.
No. I'm just wondering how this relates to, so like you're taking, these are populations of cells, of yeast cells, and you've taken out this like really important transcription factor. Things are slipping over, but like does that affect the phenotype of the yeast at all?
So it makes yeast cells grow slower.
Huh.
And some experiments in the past show that they might be more sensitive to DNA damage. So the thing is, yeah, so Spt4 is not essential, but when I remove Spt4, if I do something a little bit more damaging to transcription, cell cannot survive. So you put Spt4 and you delete another non-essential protein, they usually cannot survive. So these cells are just viable, like they have just one little energy to live, and if you damage one more thing, they just collapse. But in general, I just know that from slow growth phenotype is the most consistent throughout other studies, let's say.
And like, I guess you have the DNA sequence of all of this, right? So can you do like, can you look at if the specific sequence that is there, that is being transcribed, matters?
But yeah, I'm gonna go ahead and answer anyway. So I didn't do DNA motif analysis, and I think that's also a very interesting thing to do. What I did is I checked if the GC content of genes would change, because if there's higher GC content, it's generally thought that Pol2 movement is a little bit more difficult. But I couldn't see any correlation between this phenotype and GC content. But gene motif, I didn't look at. because also I think it's a very broad thing as well, because I know that Spt4 is important for all genes equally. I see this reaction in all genes. So because I didn't see anything specific, I didn't dig into that path.
Yeah, I guess like the first thing I would think about is like any potential secondary structures in the either the RNA transcript or the DNA that's being transcribed, and see if that is affecting.
Yeah, I mean that. I think that's a good way of thinking it. But what I see is if it exists in every gene, it's probably more general than being specific.
Right, right. But I guess it might affect like the density of the nucleosome, or not the nucleosomes, the polymerases.
Yeah. Well, I think it's still something worth seeing as well, because there is a lot of data and I have so many data to compare with.
I wish I had that though.
I can pass my data on.
Yeah, let me change my Ph.D. to transcription.
Yeah, I see Asya's hand again. But...
Ah, no, I think it just didn't disappear. No more questions, don't worry.
You're more than welcome.
Cool. Well, we're at an hour. Does anyone have any more questions? So that.
Yeah, I can ask a brief question, just very quick and general. I saw that you did your bachelor's in Turkey, and I had immediately for some reason made an assumption that maybe motivation to do this like very molecular studies came from the undergraduate degree. Is that the case? Because I know that in Turkey, and I also did bachelor's in Turkey, and that most of the biology departments are like molecular biology departments, so MBGs. Is that where motivation comes from, or you because most of the time was in this topic? Yeah,
I think so too. So my program was molecular biology, and even first day when I was there, I was like, you know, this central dogma, DNA to RNA, RNA to protein, and how this works, because it works in all organisms. And yeah, that's where my interest comes from.
Okay.
And that's the project that this is the path that I wanted to take always. But just yesterday, I checked this molecular cell, like, I don't know, maybe there's a book called The Cell. It's the textbook. And I wanted to go back and see what did they say about Spt4 and transcription. They don't even mention, because these factors are this is something new. I don't know if it is good or bad. Like, because just I think that, oh my God, I do something that textbook does not mention. Is it because it's so new, or maybe they don't care, kind of thing? But like, yeah, like Pol2 they know, nucleosomes they know, but they never put them together into context. So this is something. It definitely happened to me. Well, actually, it happened to me during meme. So I remember one of the meme coordinators circulating conference, but I was in Munich, and I got an email saying that there will be conference in Vienna in like molecular interactions, and I was like, I should go. And then I met my supervisor there, my PhD supervisor there. So it was connected with that. But yeah, I agree that like from the first day, maybe in Turkey, they also give you this path, like more molecular, basic science. Yeah.
I remember
doing that. All my friends are also doing similar. Not this, but like they also ask very fundamental questions and do transcription.
Okay.
I know Yağmur is Turkish, and she's doing, I think, like theory stuff. So.
Yeah. Yağmur is Turkish, and she's doing amazing stuff. Yeah, I think. Yeah, to me, there are, well, to be honest, the most successful undergrad students also bioinformatician now, but the majority of people are doing this kind of wet lab, molecular biology kind of stuff.
Yeah. Okay.
Not many eco-evo labs in Turkey.
There is one. There is one very cool lab again in my university.
One in the entire country.
Well, I think there should be, of course, there should be more. But
in Boğaziçi, I think they have also cool labs in environmental ecology.
Yeah. But that's the other thing. Just I kind of dropped just evolution kind of thing. Like I am interested in everything in great sense. So I'm not maybe aware of all the groups in Turkey working on this stuff.
Yeah.
There has been a lot of changes in last 6 years, which I wasn't like at home.
Yeah.
Ülkü, one last question. I know you are looking for postdocs, but do you have a general idea of what you want to do next or where you want to go?
So yes, so my partner is in Copenhagen, so that's why I look for PhDs in a specific city. That's why, like, I have a couple of options, but not in the location that I want to go. So I am at the moment like taking some interviews with some groups, and one of the I will tell you here, I don't know if it will come true, but I really want to study transcription and DNA damage together. This is also very like hot topic and very new. So I already know that I have some skills in transcription, but I want to combine it with DNA damage and like check how nucleosomes are, how chromatin environment is changed during DNA damage. This is coupled with transcription and so on. So we'll see if that will come true. But
I know that at the University of Copenhagen, they do a lot of like ancient DNA research, and part of that is looking at DNA damage.
Yeah, maybe in biology faculty. Yeah, I'm checking. Like at the moment, I think someone will be suspicious at the university because I'm checking web page of every group every day and reading through their. Like I get very like nice general background now, but yeah, there is one group doing actually I think evolutionary biology with DNA damage and stuff. Like I go through them one by one, but it's a bit difficult because it's also pandemic and people. I think they're more hesitant to invite someone because everything is on hold. I know my partner is in Copenhagen at the moment. They do half-time kind of lab work. So it's not perfect timing. But it's also, I mean, I just last month we submitted the manuscript. So that gave me a lot of time to work on and stuff. But yeah, hopefully in a couple of months, I'm going to be in, again, fundamental biology, but maybe in a different perspective, to be able to combine two different approaches later if I have a bigger project.
Yeah, there's the University of Copenhagen, and then there's the like DTU, right? Denmark Technical University. And I think they do a lot of molecular stuff. They do a lot of like synthetic stuff.
They do a lot of, they use, actually they're also super cool. They are using yeast cells as factories, and they want yeast cells to produce some chemicals, some food, like it's more biosustainability kind of projects, which is also very exciting. But I don't know, it would be quite a change in my career. But I am even considering that too, because like Meme also taught me that like I change my every 5-6 months, and I learned a lot. So if I can do it again, it will be a lot of fun.
Cool. Good note to end on. All right. Thanks a lot, Ülkü. Next week, we will have Lafi talking about something. I forget. He's here, though. So hope to see you all next week.
Emily. Sorry, I was thinking Emily. No, she hasn't responded.
See you, everyone.
And you had mentioned that you were going to try